Hello, everyone. It's Dennis again. This time we're going to get a little bit into the theory of performing extractable and leachable studies, specifically with respect to the design and use of simulation studies, well, for LVPs. You don't need to know any more about me. If you don't know me by now, then forget it. What we're going to really quickly do, one of the disadvantages of presenting remotely is I suspect many of my introductory slides are already redundant, but I can't be 100% sure. But we'll go through those really quickly in terms of, reiterating what the LVP dosage form is, what the LVP challenge is. Then we're going to get into, what a simulation study is, what a simulation study is supposed to accomplish, and how it addresses the LVP challenge. And then we get into the meat of the presentation, which is how to design an effective simulation study. Because it's one thing to know what a simulation study is. It's another thing to know how to design the simulation study. And that's important because the regulatory acceptance of a simulation study in your submission depends upon how well you can defend the design parameters. If one can effectively defend the design parameters, then a simulation study has a very important place in your regulatory file. If there is no justification, or if the justification is poor, having a simulation study in your file actually creates more problems than it solves. So it's really critical that the study be designed appropriately. The learning objectives. Again, I think the first three you've already learned by now. Maybe the concept of of simulation study needs a little bit more work. But again, I'm going to tell you, based on good science, how to select the proper simulating solvents, how to appropriately accelerate an extraction, and how to account for surface area to solution volume effects. Again, what's the issue with LVPs? It's the large volume and the complex composition that makes it difficult to screen LVPs for leachables down to the AET. This picture just tries to put things in perspective in terms of, how different the daily dose volume is for typical parental or not pharmaceutical dosage forms. I mean, a puff in an inhaler from an MDI is measured in microliters. That's how small it is. An eye drop is measured in, tenths of a milliliter. The contents of a pre-filled syringe is on the order of 1 to 10 milliliters. An LVP, now we're talking about liters. And if you market a dialysis solution, now you're in the realm of 5 to 10 to 15 to 20 liters per day. So the daily dose volume among pharmaceutical dosage forms is just orders of magnitude different. And why is it important? It's important because the dose makes the poison. One of the most time-honored foundations of toxicology is the dose makes the poison. Well, the dose is directly related to the volume of medication used. It's no more simple or no more complex than that. The larger the volume of a medication that a patient receives on a daily basis, the lower the tolerable dose. It's as simple as that. It's an inescapable relationship. The value of a safety threshold decreases in direct proportion to the increase in daily dose. You've seen a picture like this before, with a little bit less of the details. And what is added to the picture in this particular case are three cases, three examples. So let's consider an MDI where in a canister there's 200 labeled actuations, and the daily dose is 10 actuations, and the volume of the container is 0.5 mils, half a mil. In this particular case, when you work the math, the AET becomes 6 parts per million, 6 microgram per milliliter. And that's easy to accomplish with state-of-the-art analytical chemistry as it exists today. Any laboratory that you would work with can get there. Let's think of an inhalation solution, a small volume parental, where you have 3 mils of a drug product in a single dose LDPE bottle, and the dose becomes 3 bottles per day. In other words, this inhalation solution, a patient gets it three times a day. Here, the AET is now 17 parts per billion. Again, many laboratories can get down to 17 parts per billion in simple drug products, but you're beginning to push the envelope. Case number 3, an LVP, 1 liter of drug product in a bag, and it's 1 bag per day. Here, the AET without even adjustment is 0.15 parts per billion, which is practically impossible to accomplish without heroic efforts. And this deck is stacked against us with respect to heroic efforts. If this was a target compound, maybe we could get down to 15 parts per billion, but we're talking about screening a drug product at 0.15 parts per billion for unspecified compounds. That's a pretty tough analytical challenge. So it's the AET challenge is how low can you go? Can your method's LOD be below the AET? If it was sterile water, maybe. If it's a lipid emulsion, probably not. Maybe this is a better picture of what the LVP challenge is. Yes, this particular slide illustrates the reality of the LVP challenge. Think in terms of, on the left-hand side, this is the LVP challenge if you're talking about sterile water for injection. Three leachables in sterile water for injection that are easily seen because the baseline is simple. It's like sailing on a calm sea. Here's what the same chromatogram looks like if it's a 20% lipid emulsion, where these are all the impurities and degradation products of the lipid emulsion. It's a challenge. I would challenge anyone to find these same 3 peaks in this chromatogram. You're no longer sailing on the calm sea. You're sailing in the middle of a tropical storm. That's visually what the reality of the LVP challenge is. How are we going to deal with the LVP challenge? Well, we're going to stack the deck. We're going to stack the deck in our favor by changing the nature of the study. We're going to change everything about the drug product that makes it difficult to screen and turn it into a property that makes it easy and easy to screen. And we're going to call that study that makes things easy a simulation study. And we're going to call it a simulation study because that's what it's supposed to do. It's supposed to simulate what happens to your drug product while it's being stored in the container closure system. How are we going to do that? Well, the first thing about the drug product is we're going to change it. We're going to completely change the formulation of the drug product and replace it with a simulating solvent that's easier to test. So in essence, the lipid emulsion is going to become sterile water for injection. In essence, not in reality, but in essence. Okay. The second thing we're going to do is we're going to contract the calendar. The actual conditions of contact for most LVPs is a room temperature shelf life of 2 years. So instead of a study that takes 2 years to do, we're going to compress that study into a month. And maybe not that less, but that's the concept. Additionally, the third thing we're going to do is we're going to exaggerate the conditions a little bit. So we're going to take a bigger bottle and exaggerate it with a smaller bottle. And what that does is it exaggerates the surface area to solution volume ratio. And so that's how a simulation study is going to help us meet the AET challenge. We're going to stack the deck in our favor. The objective of a simulation study is because a simulation study by definition is an extractables study. As soon as you say that we're not going to use the drug product, we're not going to use shelf life, it's no longer a leachable study, it's going to be an extractable study. But instead of producing an extractables profile that's somewhat different than the leachables profile, our objective here is to produce an extractables profile that is indistinguishable from a leachables profile. That means the extractables profile contains, as extractables, every substance that you would find in a drug product as a leachable at levels that are equal to or greater than the levels of leachables in the drug product. That's what a simulation study does. It produces an extractables profile that's the worst case leachables profile. The key parameters that we need to simulate about the drug product is its composition, the temperature and duration of shelf life, and the stoichiometry. And what I mean by stoichiometry are things like surface area to solution volume ratio. All right, let's focus a little bit about solution composition. And we're going to talk about three characteristics of the solution that we need to account for in a simulation study: polarity, pH, and reactivity. I hope you like the cartoon. Polarity is essentially the concepts of like dissolves like. If you have a nonpolar substance in your container closure system, that nonpolar substance is going to accumulate as a leachable in your drug product to a greater extent if your drug product is nonpolar. The opposite is true for a polar leachable. A polar leachable is going to accumulate in a drug product to a higher concentration if the drug product is polar. So what you're trying to do is match the polarity of a simulating solvent with the polarity of your drug product. One of those two things is easy. The other is not so easy. For solvents, it's easy to determine their polarity because there are polarity tables. One can go to the internet, type in solvent polarity tables, and you can have your choice of solvent polarity tables. So it's easy to contrast the polarity of water, ethanol, and hexane. That's easy. That's easy to do. What's hard to do is what is the polarity of my drug product? Okay, you can't go and find a table that lists all drug products and their polarity. You have to establish what the polarity of your drug product is. And unfortunately, it's not as simple as putting a pH meter in a drug product and measuring a pH, because there is no such thing as a polarity meter. You can't measure polarity directly, so you have to go after it indirectly. One way to do that is to use surrogates for polarity. Things that you can actually measure. And one example of that is dielectric constant. Now, I'm not saying that dielectric constant and polarity are the same and the same thing physical chemically. They aren't. But dielectric constant and polarity can be related, so that if one measures the dielectric constant of a drug product, they can correlate to what its polarity is. So that's one way to do that. Another way to do it is the use of polarity markers. For example, this dye, this Reichardt's dye. If you put Reichardt's dye into a solvent, it will change its UV absorption characteristics depending upon the polarity of the solvent. So, this dye has the ability to conformationally change its structure in response to polarity. So again, its absorption spectra changes with respect to polarity. So you put the dye in your drug product, you put the dye in a simulating solvent, and the right simulating solvent is the one that has the same UV absorption characteristic. The other way to do it is to experimentally determine an extraction power scale. And this is an example of an extraction power scale. What an extraction power scale is you actually extract something with different simulating solvents, and you measure the concentration of an extractable. Okay, in this particular case, in this particular model, the entity that was extracted was a DEHP plasticized PVC, and the extractable that was measured was DEHP concentration. Okay, so you put this PVC resin into uh, solutions with different percents ethanol, and you measure the DEHP concentration, and you with the appropriate mathematical manipulation, you get a straight line. Okay, that's great. Now you put, for example, water per injection in contact with the same resin, measure its DEHP concentration, extrapolate over to the y-axis, and that's the percent ethanol in the appropriate simulating solvent. And, as you might guess, for water and injection, the percent ethanol in a simulating solvent is essentially zero. But if you wanted, for example, to find the right simulating solvent for 20% lipid emulsion, again, you would put the resin in the 20% lipid emulsion, let it come to equilibrium, measure the DEHP concentration right up there. That's the DEHP concentration. Go over to the y-axis, and the right simulating solvent for 20% lipid emulsion is roughly 65% ethanol. So that's just another example of how one can get at the polarity of a drug product and then pick the right simulating solvent. For most LVPs, which are aqueous formulations, it's the pH that's the most important parameter to simulate in your simulating solvent. And that's because the solubility of acid and base extractables is going to vary as a function of the pH. And acidic extractable is going to accumulate at the highest concentration at high pH. A basic extractable is going to accumulate at the highest concentration at low pH. So the amount of an acidic or basic leachable in your drug product is going to depend upon the drug product's pH. Okay, most commonly encountered acidic extractables have a pK, an acid dissociation constant of 7 or less. Most commonly encountered basic leachables have a acid dissociation constant of less than 3. Most LVP products have a pH of between 3 and 9. More acidic and more basic than that, frankly, it hurts on injection. So it's relatively rare that LVPs are formulated outside this extreme. So therefore, 2 simulating solvents, one prepared at pH 3 and one prepared at pH 9 or 10, reasonably bracket the universe of leachables and drug products. Okay, so there's the recommendation on the pH for simulating solvents for LVPs. pH of 3 and 9 will cover just about everything. All right, let's go to temperature and duration. Well, again, the concept is simple enough. The higher the temperature, the faster solvent, the faster substances diffuse out of the packaging system. It's as simple as that. We increase the temperature to decrease the duration. I'm going to suggest to you 2 means of doing that, 2 mathematical ways to determine the accelerating factor. Both are based on Arrhenius equations. The first is taken out of ASTM F1980-16. That means it's been revised 16 times since 1980. The standard guide for accelerated aging of medical devices. Okay, the concept here is an accelerated aging time is going to be equal to the actual aging time divided by a constant, this constant C. And this C depends upon the accelerating temperature, the temperature of actual storage, and again, a factor called Q10. We're used to seeing Q10 being a factor of 2. If I simplify what this expression is trying to communicate, is every 10-degree increase in temperature decreases the time by a factor of 2. That's the essence of this particular equation. The other is empirical. Empirically, it's been noticed that diffusion coefficients for common extractable substances behave in a predictable, in a simplistic manner. That's that every 20-degree increase in temperature causes a factor of 10 increase in the diffusion rate. So you have again, when you're talking about a simulating factor, it looks very similar to the ASTM. Here you just have a 10 instead of the factor of 2, and here it's a factor of 20 instead of 10. If I try to speak to this simply, this is every 20-degree increase in temperature decreases the necessary time by a factor of 10. Okay, so you have two approaches. Which one is the better? Yeah, well, I guess it depends upon your definition of better. If your definition of better is which one is more conservative, it's the ASTM one. The ASTM one always predicts a longer period of time than the factor 10, a rule. So again, in a submission to the agency, generally speaking, they encourage one to be conservative. The more conservative approach is the ASTM approach. And then, again, the mathematics is straightforward enough. If the duration of contact is 10 hours, and you go from 25 to 45 degrees centigrade using the factor 10 rule, what is the accelerated duration of extraction? Well, it's tenfold smaller. So a 10-hour contact becomes 1 hour. A 2-year, 730-day shelf life becomes 73 days. That's the calculation. It's simple and straightforward. All right, let's talk about extraction stoichiometry, surface area to solution volume ratio, or material weight to solution volume ratio. And all I want to talk about there is the fallacy. Many times you see people justify extractions based on surface area to solution volume ratio, but what they don't understand is at the same time they're changing the surface area, they're also changing the mass of the container. So an effect that they subscribe to surface area is really just a mass effect. The other fallacy, and it's really a terrible fallacy, is the assumption that if you increase the surface area to solution volume ratio by a factor of 2, the concentration of the extractable will increase by a factor of 2. It sounds great, right? Sounds simple, nice and easy. It violates every rule of physical chemistry that I can think of. That simplification is simply false. I can't say it any other way. And the reason that it is false is because the distribution of a substance between a container and the drug product is a partitioning It's a partitioning phenomenon. It depends upon the partition coefficient. And when one calculates the concentration of, in this case, an extractable in an extract, the partition coefficient is an important part of that equation. And this is what it looks like. This is what it looks like for a substance with a low partition coefficient. In other words, a substance that prefers to be in the liquid and not in the plastic. One does see an increase in concentration as the surface area increases, but it's not one-to-one. If I had drawn a one-to-one line in this particular plot, you would see that at the higher surface area, this deviates from one. So even in the best case, where an extractable loves to be in the drug product, it's not one-to-one. But in the case where the extractable likes to stay in the plastic, so for example, an antioxidant, for example, DEHP is a plasticizer, the concentration almost changes, it doesn't change at all as you increase the surface area. So the assumption that, hey, I'm going to throw 10 stoppers in a vial, and when I get the extractables result, I'm just going to divide it by 10. Forget it. That doesn't work. That is not the case. The value that you would have to divide it by depends upon the partitioning coefficient. I do want to quickly point out that maybe everyone's been talking about organic extractables. There is one elemental impurity that's a particular challenge for LVPs, and that's aluminum. And that's because of the aluminum rule. So the FDA has published an aluminum rule. And for parental nutrition products, TPN LVPs, they have to be labeled as potentially containing a toxic amount of aluminum if their concentration exceeds 25 micrograms per liter. That's 25 parts per billion. It's one of the reasons why you see parental nutrition products in plastic and not in glass anymore. It's almost impossible to put a drug product in glass and not get 25 parts per billion aluminum. So I just want to make people aware that the LVP challenge exists for at least aluminum as well as organics. This is what the labeling looks like. Who wants this labeling on their drug product? The answer is no one. No one wants to have this labeling on their drug product. So it's a really challenge to keep LVPs down to 25 parts per billion or less aluminum. Okay, so in review and in closing, a properly designed simulation study produces an extractables profile that is the worst case leachables profile. The way you do that is by properly choosing the solution composition, the temperature, duration, and the stoichiometry. For solution composition, what you're optimizing is polarity, a pH, and reactivity, the ability of the solution to actually react with your drug product. When considering temperature and duration, the ASTM, the AST method for calculating an accelerated extraction is the preferred choice because it's more conservative. And when considering stoichiometry, it's all right to exaggerate slightly. So if you exaggerate by a factor of 2, if you put 2 stoppers in a vial, that's okay. That's okay. Partitioning doesn't have that big an effect if the exaggeration is realistic. But again, I've seen people try to put 85 stoppers in a vial and claim that they can divide the number that they get for their extractables by 85. That's just not credible. The FDA, the regulatory agencies will get you on that. And lastly, but not least, be aware that the LVP challenge includes aluminum as well. some key references that if you'd like. And thank you very much. I'm more than willing to take questions.